hallucinations, while others died. And instead of saying, you know what, we took a no on this one, boys, and we should probably avoid these mushrooms from here on out, no. Someone had the brilliant idea of consuming the urine of the reindeer instead, since he believed that the urine would sterilize the toxins but keep the effects of the mushrooms intact. And he was right. And after that, humans and reindeers could go on wild hallucinogenic trips together. Some people even believe that this is how the story of Santa Claus was created. This theory is called the Magic Mushroom Christmas Theory. Number 2, Jaguars. I guess even an apex predator needs a break from killing and ripping other animals apart. They do this by seeking out the
work on dopamine led to him being awarded the Nobel Prize in Physiology and Medicine in the year 2000. If we're going to be talking about dopamine, it might be worth mentioning how it's made. Dopamine is made starting from phenylalanine in the body, which is an essential amino acid. Phenylalanine uses the enzyme phenylalanine hydroxylase, 2-PAH, to make tyrosine. Tyrosine is then converted through tyrosine hydroxylase, 6-PAH, which forms L-DOPA. L-DOPA is then able to undergo a decarboxylation from the enzyme dopa decarboxylase, which results in the formation of dopamine. So if that went over your head, just to summarize briefly, dopamine is made in the body, starting from the essential amino acid phenylalanine. After phenylalanine is converted to tyrosine, tyrosine gets hydroxylated into L-DOPA, and finally L-DOPA is decarboxylated to afford dopamine. So that's the significance. Biology, a ligand is any molecule that binds to a receptor. For the purposes of this video, we're talking about ligands from a biology perspective rather than a chemistry perspective. I apologize if that upsets any chemists out there. The general mechanism of G protein coupled receptor activation involves the following steps. Step 1, ligand binding. When a specific ligand such as dopamine or acetylcholine binds to the extracellular domain, which is just the part outside of the cell of a G protein coupled receptor, it induces a conformational change in that receptor. The second step is G protein activation. Once the G protein coupled receptor undergoes a conformational change, it enables an interaction with a heterotrimeric G protein located on the intracellular side of the membrane. This is on the inside of the cell. G proteins are composed of 3 subunits: the alpha subunit, the beta subunit, and the gamma subunit. The third step is G protein exchange. When the G protein coupled receptor is activated by binding to a ligand such as dopamine, a complex chain of events is set into motion. The GDP molecule bound to the alpha subunit of the G protein is replaced with GTP, leading to the dissociation of the alpha subunit from the beta and gamma subunits. The fourth step is effector activation.
This is
the
how you get
GTP-bound
high.
alpha subunit and the beta gamma subunit
This is what's
can now
going on in your
independently
brain when you're
regulate
getting
various
high.
intracellular
You're hitting
effector
this vape.
molecules, such as enzymes or ion channels. This just means that the GPCR is able to interact with other parts of the cell as a consequence of the interaction with the neurotransmitter or other molecules. These effectors mediate the transmission of the signal initiated by the
This is in
ligand
your
binding to the
brain.
G protein coupled receptor. The fifth step
I'm
is
doing
signal
the intro, by the
amplification
way.
and
I'm
termination.
doing
The
sample
activated
before we
alpha
talk.
subunit or the beta gamma subunits can interact with multiple effector molecules, leading to the amplification of the initial signal. The signaling cascade continues until the GTP on the alpha subunit is hydrolyzed to GTP, which is just going from the triphosphate to the diphosphate. This is where we lose energy, and I like to think about GTP as the Canadian dollar of the cell. It's not
One
used
time
as
I
much
put my
as
hand
ATP,
on that
but
rock,
it's still
it gave me
common enough.
energy.
The hydrolysis of GTP to GDP
You know what
causes
that's
the
for? It's,
reassembly of the
it's
G
a
protein
Himalayan salt
complex
lamp.
and terminates the
It's supposed to
signal.
be,
Once the GPCR
supposed to,
cascade is complete,
absorb
several different
energy
physiological
or
responses
something,
can occur,
or-
depending on
Yeah,
the
that's
type
what
of
I
G
did.
protein as well as the
I
cellular
put my
content.
hand over it
For
and I did a
example,
vision.
let's talk about the context
Nice.
of dopamine signaling. Dopamine binds to
I did
dopamine
that the
receptors,
other day.
which are a class of G protein coupled
So you put
receptors.
your hand
These
over it and then
receptors
you
can
had
be
a
broadly
vision.
classified into D1-like receptors, which includes D1 and D5 receptors, in addition to D2-like receptors, which includes D2, D3, and D4. When dopamine binds to D1-like receptors, these GPCRs couple to G alpha subunit proteins